So I'll tell you more about the work that we do at UCLA, academic work that we do with our team, students, and postdocs, and how we have been commercializing or licensing out some of these technologies. Some of them are related to mobile health, but some of them are a little bit maybe outside of mobile health in terms of the exact core. But hopefully you will get the idea that these are all at the intersection of computational imaging, sensing, and the use of light for measurement tools. In general, my lab is interested from the very beginning at the intersection of imaging and sensing, and how computation could simplify instrumentation, to democratize instrumentation that we use in our labs today. And the first generations of devices that we created utilize the smartphone as the basis of advanced measurements. For example, these are some of the earlier prototypes that we had to convert the smartphone into a high-end microscope to the level where you could see an individual bacteria, DNA molecule even, that's labeled with intercalating dyes, and size them with about a kilobase pair sizing accuracy. On top of that, we are also interested in sensing, and we created interfaces that basically not only converted the smartphone into a microscope, but also into a sensor of various different kinds. All in all, we would like to perform the same measurements, the same images that you create in diffraction-limited benchtop counterparts with basically inexpensive and field portable interfaces that could work in resource-limited settings, in field settings, or at the home for looking at specimen with specificity, sensitivity, resolution that you expect from their lab-grade counterparts. For example, just to highlight some of the earlier work from our lab, this is a fluorescence microscope that is being installed at the back of a cell phone. So the cell phone camera, this is a Windows phone from maybe 10 years old now, and with very inexpensive attachment to the smartphone, which can be detached if you don't want to use it as a microscope, we were able to see individual nanoparticles and size these particles, and also bring some specificity with fluorescent tagging. This is actually human cytomegaloviruses that we are imaging with a mobile phone-based microscope. This is the cell phone's image showing individual particles of individual viruses that have been labeled with fluorophores and imaged with the smartphone camera interface. Inexpensive, rapid, has specificity, and on the right here, I'm giving you an expensive confocal microscope image of the same field of view to give you the idea that really you're competing with very expensive benchtop instrumentation and democratizing access to some of these traditionally bulky and expensive benchtop instrumentation that we have in our labs. A similar device is shown here. This is with a very simple silane chemistry. You can stretch DNA on a glass slide and image it with a mobile phone-based microscope. This is the cell phone's image. If you zoom in, you would be seeing these stretched DNA molecules. These are labeled with fluorophores that let you see and size individual DNA molecules to the level where you can actually look into copy number variations in the genome and look into, for example, drug resistance of TB and malaria through these kinds of probes that are specifically labeling certain fragments of the genetic material of, for example, some of these pathogens. It's come a long way. We've created multimodal mobile microscopes where within the same embodiment, you can have bright field microscopy and fluorescence microscopy at two different channels, which means you can take a tissue specimen and, for example, label two different target biomarkers with one fluorescence, let's say green, the other one red, and start doing multiplexed analysis. In fact, in this work in collaboration with a Swedish group, we used rolling circular amplification process to look at basically targeted DNA sequencing at the tissue level where mutant labels were forming clusters of DNA labeled with basically 1 fluorophore, let's say green, and the wild type was labeled with red. So you could do bright field imaging of tissue sections and show targeted sequencing. We demonstrated the efficacy of this platform with colon cancer patients to show that you can do DNA sequencing at the and find out as single nucleotide level differences through the readout coming from these mobile microscopes. This is a multimodal example of a mobile microscope installed on a smartphone in an inexpensive and field portable manner. In addition to creating interfaces that go at the back of a smartphone camera to convert it into a high-end fluorescence or bright field or multimodal microscope, we sometimes also created standalone devices empowered by economies of scale. Taking the CMOS imager, these beautiful 10 megapixel, 20 megapixel imagers at the back of your cell phone, they're actually great for building standalone microscopes. For example, this one is a computational microscope that is powered by a CMOS imager from a cell phone camera, and it can reach diffraction limited resolution across a very large field of view on the order of 20, 30 millimeters square, and perform beautiful imaging of specimen, especially transmissive specimen, even in field settings. If you look inside one of these computational microscopes, you see that actually it's quite different than a traditional light microscope design. These are holographic on-chip microscopes, and they're different in terms of their working mechanisms compared to traditional lens-based microscopes. If you look inside one of these devices, there's no lens. It's actually a lens-free, lensless microscope. The main component is a CMOS imager. Think of this as the 10 megapixel, 20 megapixel silicon chip with millions of pixels inside, which gives us essentially an image. However, there's no lens in here, and the sample goes right in front of it. Basically, we're detecting the holographic shadows of the specimen captured by the CMOS imager. This front end here that I'm pointing to, that's actually an illumination unit composed of light emitting diodes, very inexpensive. You have something like 20 LEDs in here. Very, very inexpensive, a few dollars to put together. They're individually turned on and off to give you more information. We call this pixel super resolution. Stated differently, if you give me a 10 megapixel imager, I make it 1,000 megapixel so that I can actually improve the resolution over the same field of view. This framework is a computational framework, and it's called pixel super resolution. Think of it like a security camera. If you look at one of these frames of a security camera that is low resolution, you cannot resolve the fine features of the specimen. But if your aperture, your hand, or the sample itself is moving slightly left and right, like jittering, you can actually take the sequence of images, let's say 10 frames, and synthesize a super resolved image where you divide individual pixels that undersample your image into subpixels. That's the gist of this framework that we're using here, except we're applying this framework to holography to create higher resolution holograms that can look at specimen in field portable instrumentation. So the idea here is we do this pixel super resolution to reveal finer features of the holograms of specimen. The end result is you have higher resolution and you can reveal features that would normally be washed away. For example, this object is a synthetic object that we created to quantify resolution. If you have a standard CMOS imager off of your cell phone, it cannot be imaged in a lens-free on-chip microscope because of the pixel limitation. By doing pixel super resolution rapidly, we can resolve it beautifully well within the same active area, within the same CMOS imager, all done computationally in a lens-free manner. That means we can now start to bring together an inexpensive prototype like this. It's as tall as an iPhone, maybe 200 grams in total weight, controlled by a laptop, and it can look at transmissive specimen with a very good resolution due to pixel super resolution framework and very large field of view. We've been using it for various different specimens for telepathology applications, for diagnostics. I don't have time to go over all of these. I'll give you some small set of what we've done with it. One of the first things that we looked into it using these kinds of microscopes was thin smear. So this is actually a malaria-infected thin smear where there's a monolayer of red blood cells mostly. Normally, a traditional microscope would be scanning the field of view to find these infected cells. It's the red blood cell, and this Giemsa-stained purple-looking thing here, that's actually the signature of the parasite, Plasmodium falciparum. In our lens-free microscopes, we get synthetic data and reconstruct them rapidly these days, mostly using neural networks to give you a beautiful contrast. But it's not making microscopy great again. It's just not making it simple and cost-effective. It's also higher throughput, meaning that we can look at larger areas and find these infected cells and look at the condition of the patient. For malaria, microscopy is the gold standard for tracking of the infection and counting peristemia. It's very important to see if you have drug resistance issues, etc. Besides diagnostics, it's also important for understanding therapy. And one of the major advantages that I mentioned is the imaging field of view. So this tiny chip here, that's literally used in our cell phones. Of course, this is an older version, but newer versions are about the same, similar size. This green-looking active area, that's where you have millions of pixels. That's where we place our samples. And the samples give you a very large field of view, in this case, 20 millimeters square. This is huge compared to traditional light microscopy, which would be typically looking at sub-millimeter square fields of view. That means we can look at large specimen without scanning. This is a histology section. This is breast tissue section, 3 microns thick approximately. A diagnostician to reveal the subcellular features and to diagnose all kinds of conditions would need to look at these kinds of images, but they would scan with a, let's say, 40x objective, which has this field of view, and capture a few hundred of these fields of view to give you a whole slide image. We can do this much higher throughput because this is on chip microscopy where the entire active area of the chip is our field of view. And the advantage of the field of view is highlighted here. This is pneumonia H&E whole slide, which is, this image is created by lens-free on-chip microscopy with different colors of LEDs working at the same time. Some beautiful math to get these kinds of images reconstructed, and over such a large field of view. Right, this one is about 30 millimeters square active area of another kind of a CMOS. You can zoom in and reveal various different features as you zoom in, coming into focus computationally and revealing basically the subcellular features. To give you an idea that these are not just toy microscopes. Yes, they're small, they're cost effective. It would cost us less than $100 to put together one of these devices. They're as tall as an iPhone, but they are diffraction limited, beautiful microscopic solutions for spatially looking into specimens, pap smears, blood smears, and urine samples, tissue samples, you name it. One of the scientifically interesting aspects of lens-free on-chip microscopy is that field of view or sample volume is not tied to resolution, which means I can use different kinds of sensors and create extremely large fields of view. Everything that I've shown you so far had a traditional CMOS taken from a mobile phone. That's why the field of view was kind of like this, 20, 30 millimeters square. But this one here has a different scale bar. It has a centimeter scale bar. It's got actually an active area of an 18 centimeters square CCD. This is a charge-coupled device where you're seeing reconstructions of human sperm flagellum. Submicron features are revealed, and this has about 1.5 billion useful pixels to it. Depending on how you want to do imaging, what kind of resolution, what kind of field of view, lens-free microscopy is a black box that gives you quite a bit of opportunities for field portable or home-based microscopic investigation of various different specimens. And over the last perhaps 10 years or so, these devices have been empowered by deep neural networks so that their reconstructions, their image enhancement, their resolution enhancement, depth of field enhancement has been now switched to deep neural networks, helping us to improve the performance of these systems. And we have been using deep learning specifically for image reconstruction tasks, image transformation tasks, not classification, not segmentation, not trying to understand if you have disease A or B, but more how to make these microscopes better, faster in terms of how they reveal these nanoscopic or microscopic features of specimen with field portable implementations. And for this task, neural networks are wonderful because training takes time, depending on availability of samples. Training can take maybe a couple of days to a couple of weeks, depending on how you digitize samples. But post-training inference is non-iterative and extremely rapid. And that's the biggest advantage of using neural networks for image reconstruction, because it's opening up new avenues for us to empower our mobile microscopy solutions. In fact, we've been creating some very powerful solutions for microscopy and sensing using deep neural networks, reconstructing some of these holograms that I've described, so that we can do high throughput screening of extremely large sample volumes, whether it's air, bodily fluids, water, and create systems that can look at micro and nanoscale objects at extreme throughputs that is basically second to none in terms of measurement science. So one example is shown here. This is a bioaerosol sensor. It's based on lens-free holographic microscopy and deep learning working together. So as you can see, it's very lightweight, maybe 250 grams in total, and battery operated, and slightly taller than an iPhone. What you see here is a lensless microscope at the heart. So right here behind this closure, you have a lens-free microscope, exactly what I've described to you in the previous few slides. The difference here is that this is specializing particles in air. This is an inlet nozzle that takes in 13 liters of air per minute, rapidly sucks it up, and basically deposits the particles on a transparent specimen, transparent substrate, let's say a polymer or a glass. Before and after you take in the air, you capture images to understand what you captured in that sampling. So within just 30 seconds or 1 minute, you've sampled something like 7 to 14 liters of air, and you're looking into reconstructions of what you've imaged to understand what you have in your air that you're breathing in. So this is a label-free bioaerosol sensor based on lens-free microscopy, especially important for looking into, for example, mold particles, mold spores, or pollen particles with specificity and sensitivity coming from basically multicolor imaging. Holography is very powerful here. It can not only generate these kinds of beautiful images, these are pollen particles that are imaged by this device, captured on a substrate, but you can also not only get the morphology sufficiently rich to classify them using a convolutional neural net, but also look into color channels for red, green, and blue illumination to bring more specificity through the absorption processes of these particles. We've shown more than 94% classification without any labels for black mold, for different kinds of pollen particles, showing you that it has actually sufficiently rich resolution and spectral signatures for label-free characterization. Normally, if you want to do, for example, black mold characterization, you will have to send a technician to the site. Let's say there's a flooding event in a building and you're suspecting of some sort of a mold growth. You have to send someone and they will take samples and culture them. After a week, you will get your result. Here, it's all label-free, and you bring some initial screening tools to look for these particles, which also has obviously applications for allergen detection at the home because of the specificity and the label-free operation of these devices.
So Dr. Ozcan, real quick, wanted to ask you, since we just had FDA a little bit earlier, I'm just wondering now, in terms of approval for some of these things, like something like this has greater than 94% accuracy. Just wondering, is this something intended for people to use and then then you get more of a ground truth, or is this intended to actually replace the state of the art?
There is nothing at the home that can measure pollen particles specifically and sensitively right now. There are obviously particle counters like PM monitoring devices that are based on light scattering. There's no intellectual property left on that technology because it's a very old technology. What they do is shine light through particles, and the scattering cross section can be used to size particles approximately. But those kinds of approaches would not work for big particles. A pollen particle is anywhere from a couple of tens of microns to more than 100 microns, irregularly shaped and with different kinds of contrast mechanisms. So today, there is nothing that can look into mold, spores, or pollen specifically and sensitively, and that's why mobile microscopy is necessary. Second, high throughput is necessary. You need to look at very large volumes of air rapidly, and that's why the nozzle and the impactor here is very important. It's not just a microscopy problem, it's also a sampling problem. This device can sample about 13, 14 liters per minute if you want. And then you have a huge sensitivity because of that. In terms of the FDA, this is a measurement device. Microscopes are not FDA regulated. Unless they are used to sense specimen or provide specific information that diagnosticians use to diagnose the patient or a condition. Here, the intent is to use them as blanket sensor. It can give you concentration of, for example, pollens of interest. Let's say you're suspecting in California, let's say, 10 different pollen particles being the most common allergens. With some good specificity and sensitivity, we can build classifiers for all of these based on these kinds of microscopic images and report at the home for certain patient populations, density of these particles, and depending on that, regulate the intensity and the frequency of HVAC that they use, or the filters that they use, or medication that they use. Depending on who's using it for what purpose, there might be different levels of regulations, but in general, microscopes are not FDA regulated until they hit that junction of being used for specific tests. Even in the case of certain lab developed tests, they're outside of the FDA regulation, like lab developed tests, LDTs, that are frequently used in clinical microbiology labs.
Wonderful, wonderful, thanks. Some of your other work, though, does touch on more health related.
Absolutely. There is FDA angle in some of the later work. Maybe in the second half of my presentation, I'll touch on some other technologies where FDA landscape is a little more convoluted.
Thank you.
A sister of this technology is actually shown here. This is a lens-free holographic cytometer. Replace air that you've seen here with water and continuous flow, which means this device is a cytometer, but it's not your father's cytometer. A typical cytometer today would be like $100,000, and it would easily take a good, desk space. Consider a medium-sized desk. That's where a cytometer would sit. This is actually democratizing cytometers, and this is actually an imaging flow cytometer, which means it's not just giving you count of certain cells. It's providing you beautiful microscopic images of specimen as they're flowing without stopping the flow. Think of a throughput of something like 0.1 to 0.5 liter per hour. Huge, right? For microscopic imaging, 100 mil, 500 mil. This is huge. A typical microscope would be looking at tens or maybe a few hundred nanoliters. We're looking at here hundreds of milliliters with a throughput that is second to none. And this system is also mobile, inexpensive, it's shoebox, and in real time it creates images of these are plankton from the Los Angeles coastal line. Beautiful color. This is actually coming from a lens-free multicolor holographic microscope inside this device. Every one of these objects, when they were imaged, it was floating something like 1 to 2 centimeters per second, so rapidly flowing at the microscale, obviously, and we were pulsing the illumination to create these beautiful images that can be used for recognition. So this actually is a product. I started up a company in 2016. This is also my conflicts of interest disclosure. Any company that I'm referring to here that I co-founded, I have financial interest to disclose. So I established, co-founded Lucende Inc. in 2016 to commercialize imaging flow cytometers that we built in our lab to look into micro objects in liquids in a high throughput, high resolution, and cost-effective, inexpensive, and portable manner. So Lucende is obviously looking into a portfolio of different problems where cytometers or particles in water or liquids in general need to be measured. So all of these different market segments, they rely on microscopes for image analysis. And an imaging flow cytometer is wonderful because it provides microscopic image content and extreme throughput together in a handheld format. If you look at today, what happens in some of these domains that are microscopes, as you see benched up, the problem with these kinds of microscopes, for a given image, you're only looking at maybe 100 nanoliters. They're good, they're beautiful resolution, 100 nanometer, 200 nanometer resolution, but 100 nanoliter will not cut it for high throughput screening. So this will never work as a high throughput water analysis system, no matter what you do. If you look at flow cytometers, they are beautiful, they're fast, but they're expensive, several tens of thousands of dollars. They're not mobile. They're fairly expensive and heavy and difficult to operate. And their dynamic range is very limited. If you have an irregular, let's say irregular stem cell, good luck. It's going to clog your cytometer. I had so many complaints from stem cell researchers how they spend the entire day on a cytometer to make it work for their stem cells. So all of those problems go away with lens-free microscopic cytometry because we can look into specimen all the way from about a micron size to a millimeter size. So we have a huge dynamic range. We don't get clogged. It's a couple of pounds, and it gives you an enormous throughput all the way from, as I said, a few hundred milliliters. You can even run it to a larger throughput of, let's say, a couple of liters per hour with some resolution loss. And this is the current prototype that Lucende has. In the next, I'm sharing some movies of the product. So you can actually, it's in a backpack. If you want to look at microplastics in a well or algae, plankton in water, where you have your fish culturing, let's say, for aquaculture purposes, this is a great solution. It can run off of a laptop as shown here. What you see here is that it's rapidly sucking up some liquid from these test tubes. And what you see on the laptop's screen, that's real-time quantification of all these images. So whatever it's creating, as I speak right now, those are microscopic images of flowing particles in real time. And this is essentially a shoebox that's working. You can also do it with tablet in this mobile version. So this is the same idea. Whatever you see there with the tiny images popping up, those are cropped images that were flowing within that liquid all the way from a couple of micron to millimeter. It's great for microplastics. It's great for looking into toxic algae. One of the areas that we're pushing this platform to make an impact as a product and as a service is actually looking into toxic algae. It's a huge problem because of global warming. Algae growth patterns are becoming more frequent and more rapid. As a result, fishermen are losing their crop rapidly. For example, right before COVID, Florida in one summer, in a couple of weeks, lost $120, $130 million because of algae blooms, toxic algae blooms. In, I think, 2016, Chile lost about $0.7 billion in a summer because of, again, the same toxic algae bloom. So you need higher throughput, you need sensitive and ideally chemistry-free, label-free imaging modalities that can constantly sample the water. And if you sense an increase in algae population of interest to you, you may treat them with some chemicals or move away your crop to somewhere else so that you can actually protect losses. Another area that we're looking into is algae biofuel quality control. So this is one of our devices that is monitoring a pool, which is looking into lipid content of these biofuel production lines using algae. It's very important because if you put a reactor there and try to kind of create biofuel, hey, some of the microorganisms might kill your algae, and it's a very delicate balance where at the micro scale, you need to constantly monitor both the health and the quality of our algae. So that's where these kinds of autonomous systems that periodically look at the water content at high throughput is essential. It's very important. And again, because of the irregular size of algae, you can't just use a standard cytometer. You must use something that never clogs. Our system can handle a couple of millimeters of particle size. Speaking of which, perhaps arguably one of the most important, depressingly urgent applications of our technology is looking into microparticles, microplastics, especially in water. Microplastics are everywhere. Our fish is feeding on it, and it's a huge problem, right? So, and these are irregularly shaped polymer particles that have some birefringence on them as well, some fluorescence properties. That's where high throughput cytometry would be also very important to quantify microplastic concentration in basically your washing machine when it's communicating with the sewage system or your water pipeline, if there's something growing there, etc. So these were examples of mobile microscopy systems that were actually empowered by deep learning, where the reconstructions got faster, better, with neural networks helping us to take some of these devices and in real time give you actually microscopic images and quantification of the type of microscopic image that we're detecting. So both classification and also image reconstruction merge together so that you bring mobility to your analysis. If you look at the heart of these academic works from our lab, they were approximating certain physical transformations. So a neural net doing image reconstruction for you, you can think of it as solving an inverse problem that has a known physical forward model. In other words, here at this baseline success story that I just described over the last several slides, this is mimicking a physics-based forward model that we were inverting. And neural networks were learning that transformation guided by physics-based reconstruction methods. So what we learned could be done using standard methods, but probably much slower than what we can do with neural networks. That was the basic story. But soon we realized that actually the opportunity landscape is much bigger than that, which means through supervised learning, you can actually bring some additional insights to microscopic analysis. And we can approximate through supervised learning and image data unknown functions. Imagine a certain function that you want to implement, but you don't have a physical understanding of this function in terms of inputs and outputs that you want, how they are related. You don't have the physical forward model. You don't know if that function can be actually implemented. Is it just a dream or is it just physically possible? Let's say you don't know that. That's exactly the type of function that supervised learning can help. At this point, I've lost some of you, and you may be wondering, who cares? Well, let me give you a little bit more context to it so that I'll bring it to something that's very important and perhaps that's touching some of our lives if you are giving a biopsy, for example. So this transformation that I just described with peculiar and abstract terms, let's say it wants to take a fluorescence microscopic image and transform it into a bright field microscopic image. So this kind of a transformation is really outside of our understanding. I cannot relate fluorescence to bright field contrast. On top of that, The sample is going through a transformation, labeling process. At the inputs, they're label-free. At the output, I want them to be labeled. So, hey, this is one of those really difficult to model or understand functions that I want to approximate. Then, if I could really approximate this function, I could perhaps really make use of it in biomedical settings. And that's exactly what we've done, because this function F that I'm just describing on the page right now will change the way that we do histology, we do histopathological staining. So for the last literally 150 years, we are taking biopsies from patients and they're stained. They're chemically stained by chromophores, because native tissue without staining under a bright field microscope, it doesn't have contrast. It looks like glass. Diagnosticians cannot understand what's going on at the biopsy. That's the reason why we involve chromophores of various different kinds developed over the last century plus to bring contrast to your tissue, so that with a microscopic image at the subcellular level, a diagnostician can diagnose all kinds of cancer is diagnosed through this histochemical staining process. This is toxic, this is destructive, this is slow, this is expensive, and this is not very repeatable. Well, that's where we are with histology. What if we could actually, through this function that I peculiarly introduced, what if this function could approximate this entire chemical process of labeling? Then I could take, instead of chemistry, a label-free image with an autofluorescence channel, look at these grayscale diffraction-limited images and transform them into the same images that come out of histology labs after their staining, but there's no staining here. Everything is virtual. We call this virtual tissue staining. Replace chemistry with a neural network to mimic what chemistry does. So here, essentially, it's cross-modality image transformations and staining of tissue. The art behind this is convolutional neural nets that fight with each other. So we're using AI to train AI in a generative adversarial network framework. But this is not one of those hallucination networks. This is actually faithful to the micro and nanostructure of tissue. If you feed noise to the generator, you get garbage. Garbage in, garbage out. But on the contrary, here, we feed in diffraction limited, beautiful 200 nanometer resolution images of the native tissue microstructure, nanostructure. This structural information here, coming from a beautiful microscope, regulates the generator so it doesn't hallucinate. It doesn't hallucinate new patients. It actually creates the virtually stained version of the actual tissue specimen from that biopsy of the patient. Everything in the next few slides I'll share with you is a new patient specimen, never seen by the training. So this is generalization of the network. Does it work with new patients? That's what we're trying to ask here. This is an H&E stain slide. H&E is one of the most frequently used stains in histology. More than 90% of all biopsies are labeled with H&E. A good H&E takes about 2 hours by a human histotechnologist to stain. We can do this in less than a minute. This is contrast enhanced, the autofluorescence signatures of the tissue samples, contrast enhanced for your eye, beautifully transformed into a virtual H&E without chemistry, without histotechnologists working on it, nicely matching what comes out of histology, except it's much faster and much more repeatable and consistent. It doesn't only work for a simple stain like H&E, it works for special stains, it works for immunohistochemical stains. This is a special stain, Jones silver stain. It's 10 times more expensive than H&E. At UCLA, this is at least 10 times more expensive than what we charge to insurance. It takes about 3 hours for a human to stain these. It's a tedious process under a microscope, very much like a chemistry experiment. Whereas we don't have any chemistry, we're just looking at autofluorescence contrast. The neural network is learning what the technologists are doing at an abstract scale, and then painting it same way as what comes out of histology lab, bypassing the entire histochemical staining process. Some new patient specimen, liver, lung tissue. Another type of stain. This is also 10 times more expensive and about 3 hours to stain. You match it beautifully well. In fact, it also works for antibody antigen specific stain. So this is immunohistochemical stain. HER2, some of you might know, it's a key biomarker for breast cancer patients. So it determines basically the therapy, so the target of therapy that the patient has to go through. And that's why for positive patients, cancer patients, their biopsy specimen will be labeled with HER2 specific antigen specific antibodies. For example, this one is a patient that overexpresses HER2. It's a surface binding receptor. The brown here is showing the expression patterns of HER2. This is a HER2 0. You don't see the overexpression here. It's almost free from HER2. This kind of a stain takes 24 hours for humans to perform. Because of the antibody antigen specificity, it's a tedious and very expensive stain. We can do this also with virtual staining using autofluorescence channels. It's more challenging for virtual staining to approximate because a molecularly specific stain. That's why we were using here 4 different stains, 4 different, autofluorescence channels. We were looking at filters from like FITC, Texas Red Sci-Fi, and DAPI to create basically an autofluorescence image cube, so to speak, as the input for these virtual staining results. In fact, we're better than the ground truth here because 30% of these stains coming from histology labs fail. We're more consistent. Because we filter out those rejection, our training is converging to only the successfully stained ones. And as a result, if you run it blindly, we're actually better. We don't waste tissue. We don't mess up. The 30% of the failure is gone. That is traditionally happening in every lab. In Southern California, for example, pick up your favorite histology lab. If you send 100 specimens, you can only use 70 of them. The remaining 30, they're garbage. We know this because we've been working with these labs for a long time now. These were examples of, hey, we generate virtual stains and it comes from histology lab. How do they look? Side-by-side comparison, resolution, color distances, structural similarity index, you name it. But the real truth is, hey, can you give these images to a group of board-certified pathologists and ask them to diagnose the condition of the patient, to score the quality of the patient? So we've created many tables like this where we asked a group of pathologists to, without knowing which one is which one is histochemically stained, which one is virtually stained, they were asked to look at the quality of the stains, nuclear details, cytoplasmic detail, etc. And the end result is this: there is statistically no discordance between our images and their quality, their diagnostic accuracy versus the traditional workflow coming from histology labs. The same is true for various different conditions diagnosed by a group of pathologists. That means not only like they're looking realistic, they're also preserving the diagnostic features of the tissue for human pathologists to act on. But it's not just matching what comes out of histology without chemistry. It's actually improving over it. The first improvement that I want to emphasize with virtual staining is that we standardize. Deep learning-based virtual staining is highly consistent. So look at, for example, these images. These are coming from UCLA Health. Basically, you see quite a bit of variation. And this is normal. And this is a problem because machine learning will not like this variation downstream. And more importantly, human diagnosticians also do not like this kind of variation. That's where neural networks shine. They're more consistent in their inference. Arguably, the biggest advantage is actually multiplexing. From a single cross-section of a label-free section, we can generate a panel of stains. And these are nanoscopically registered, which means the diagnostician, rather than waiting for some special stains to arrive from the lab, can on demand generate from a single cross-section any type of stain that they want to see. Today, this is done in a destructive way. You start with H&E because insurance covers that and it's cheaper. Then you go to the next stains and there is a time delay in between. And that is sometimes very important to avoid. And that's where multiplex staining at the same cross-section, without any chemistry, without any time delay, would be very important for improving diagnostics. We've also extended this work to biopsy-free virtual staining. So this is a dermatology clinic using a reflectance confocal microscope, looking at the skin, trying to understand, should I take a biopsy here or not? This is actually an FDA-cleared device. We've shown that you can bring virtual contrast of H&E by capturing a sequence of images from the skin with a couple of micron axial spacing, all the way down to dermal, epidermal junctions, superficial dermis layers, to give the contrast that pathologists, dermatopathologists, are accustomed to seeing, as opposed to the grayscale images of reflectance confocal microscope. And in fact, it works beautifully well. These are new patient specimen, raw data coming from the reflectance confocal microscope in vivo without taking any biopsy out. They're shown here. Pathologists are not accustomed to looking at this kind of a contrast. Nobody knows what's going on. We rapidly generate the H&E equivalent images. And you see two different modes of the device here. They're both reflectance confocal microscopes. This one is what we trained with. It's an FDA cleared version. And this one is a handheld mode. It's the same RCM, but the optics is different here. It's handheld. The model that was trained here, not only the patient here is new, never seen before, optics is also never seen before. It's showing you a beautiful generalization from device to device. In general, this is rare. Every time you change your device, you have to train or do some transfer learning. This is showing you an extreme resilience of the model from device to device, most likely because of the volumetric imaging stack that we acquire at the input of this neural network. To summarize, deep learning is really helping us for changing the way that we do histological staining, tissue staining. It's cheaper, saves labor, time. Toxic reagents are eliminated. We can preserve tissue. No need for a secondary biopsy most often, which is because we deplete tissue. And we can do multiplexing in a standardized manner. In fact, this is something that is not just an academic exercise. In 2019, I co-founded Pictor Labs to commercialize this technology. Pictor in Latin means painter. So this company is painting microstructure of tissue. So we raised about 19 million so far, and we have a size of about 25 in Santa Monica, commercializing this. And this analogy here is exactly what we want to achieve. Today's digital pathology is like Blockbuster. Every time you want to see content, you need the physical copy of the device or the storage. Same with tissue. Digital pathology today is really at its infancy in terms of how these stains are accessed. We want to be Netflix, where on demand, the diagnostician will be able to look at the same patient specimen from a single set of autofluorescence, on demand be able to generate multiplexed stains. It's exactly shown here with this movie where we're taking tissue whole slide image, autofluorescence images shown here. It's rapidly virtually stained, first with H&E. This is really a very good, beautiful H&E that you can zoom in. If you switch, if you decide at this point to switch contrast, you can bring immunohistochemical stain. This is PEN-CK IHC, which would normally take 24 hours. You can zoom out, go back to H&E on the same cross-section, and somewhere else, let's say you want a third stain, which would take a couple of hours for the histology lab to stain on a different cross-section, you can bring that. This is Mason's trichome stain on top of H&E and IHC, PEN-CK. So this is Pictor Labs, and we're excited about its prospects and how we can change the way that we do histology. In the last couple of minutes, I'll finish off with sensing. Everything that I've talked so far involved microscopy imaging and how we've been creating smart interfaces for whether microscale objects with liquids or air or tissue specimen, how to image them, how to use AI to bring better contrast to them. On top of all of that, I'm very much interested in sensing, and we've created lots of different interfaces that utilize the smartphone as a sensor. This is an earlier generation of an E. coli sensor that used immunochemistry to basically label and detect E. coli particles within milk, within water samples, a few CFU, colony-forming unit per mil level of detection sensitivity achieved. Some other examples are shown here. This one is a valve plate reader. All these 96 valve plates require some form of reader. We convert the smartphone into a beautiful colorimetric or fluorometric reader for ELISA plates to do IgG, IgM assays in a high throughput manner and use the phone itself as a quantifying element with its own algorithm. This one on the left is one of our first products, actually, that we commercialized. This one is a rapid diagnostic test reader. This is like about 15 years ago that we demonstrated that smartphones can be used for reading and quantifying rapid diagnostic tests. Colorimetric tests, chemiluminescent tests, fluorescent tests could be read with the smartphone camera and with an app quantified. So in 2009, about 15 years ago, I co-founded Salmic. Initially, it was called Holomic, to provide digital assay solutions using mobile phones. Basically, the idea was to use the smartphone, the enclosure around it, and the app to do better than what the human eye could do. A lot of these pregnancy tests or lateral flow tests or paper-based tests, they're simple, but they need to be, at that time, there was a huge push to do multiplex sensing and to do sub-nanogram per mil level of detection limit so that you quantify difficult to sense and low concentration biomarkers. And we thought the smartphone was the best platform for it because it's a universal reader, which means if you have, let's say, 20 different rapid diagnostic tests that you administer, instead of buying 20 different readers for them, the smartphone was the universal reader for it. And it was quantitative. You could go down to sub-nanogram per mil with AI, with machine learning, helping you to understand the line patterns. And you could have basically a single reader serve your needs with more sensitivity than the human eye and in a quantitative and multiplex manner. So this was, as I said, we started in 2009. Initially, this was actually funded quite a bit through various different SBIR funds, phase 1, phase 2, all the way from NIH to NSF, the Army, and it grew large. And in fact, in 2015, we've got the Technology Pioneer Award at the World Economic Forum for the impact of these mobile diagnostic tools running off of smartphones. Arguably, one of the most important products of Salmic over the last decade or so was actually a test developer software. So we had a cell phone-based device, and we had a dongle here which had its own optics, where the cell phone was just the application and the screen. So depending on the user and what they want to do, you could either have your cell phone do the imaging or a separate optical interface do the imaging and sensing, where the cell phone is basically just a display. From the FDA point of view, this was the solution that most people wanted to do, because this device, although cost-effective and using the cell phone both as the app and the camera interface, it had issues with the FDA approval process for your rapid diagnostic test, because as the cell phone's model changes, the software, hardware changes, that created problems for using your phone as a long-term product. Whereas here, the hardware itself is beautiful, and you lock it, and it never changes. And this is only a tablet for you to kind of look at and read the results. It's just basically a display. So a lot of our solutions from the mobile phone initially went into these kinds of devices, which were universal readers for all kinds of tests. And the rule of the game here was literally how to bundle all kinds of tests so that you can read them and quantify them with the same reader. And some of our customers were actually test developers. A lot of the test developers, as they create a panel of tests, they need to do quality assurance. have to create a reader that can read everything that they generate today and tomorrow. And that's why, in fact, this was a very popular product where test developers used our software to understand the fluctuations or changes in their quality assurance and in their production pipeline to regulate it. And with that regulation in mind, with those variations in mind, how to create a reader and the algorithm. So it was kind of a push-pull between our readout and their pipeline. So it was all integrated with the backend software for electronic health record systems, because these were tests that were administered in some healthcare settings, depending on the hospital and depending on how what kind of test they do. We were programming our readers to work for their test and integrate in a secure manner with their backend. In 2018, this was actually acquired by another company, and it's actually in production. Some of these readers are used. In fact, during COVID, some of your tests were read, at least in some parts of the world, through these kinds of mobile readers that we created. So I'll stop here just to summarize computation and computational imaging and sensing, all the way from creating mobile microscopes, mobile sensors, some of which were installed on a smartphone, some of which are standalone devices that are benefiting from computational imaging and deep learning, have really come a long way. 20 years ago, when we first started this line of research, with these ideas, you could barely see red blood cells, like 7-8 microns in diameter. But over the last 15, 20 years, we've seen sensitivity down to 20, 30 nanometer particles to be detected, sized, and with fluorescence specificity to single level viruses. And with the advances in deep learning and cross-modality transformations, you get quite a bit of contrast available from these mobile devices, whether it's looking at particles in air, liquids of all kinds, or even at the tissue level, subcellular level. So that's what I have. Would be happy to answer any questions. Thank you very much.
Thank you so much, Dr. Ozcan. That was wonderful. Want to open it up? Anyone? I know you do have to get to class, so we have limited time. Anyone have any questions? And feel free to unmute yourself. I have a follow-up question regarding the rapid diagnostic testing. So my first one, sort of, how do you get insurance now to cover this? It seems like it is being used now in medical settings, and also what's next when it comes to sort of, is it continuous monitoring or invasive sensing? What's next when it comes to rapid diagnostic testing?
So from the perspective of the reader, who's going to pay for the test? It's the same. So these tests are prescribed depending on the condition. Some of them are reimbursed, some of them are not, depending on, again, the patient and your insurance and what those tests are looking for, right? From the reader point of view, it doesn't matter. So let's say there are certain tests that are FDA approved or that are going through the FDA. The idea here was the test developer, the test manufacturer, if they want to have a reader for quantitative analysis, they would go through the FDA approval of their biomarker detector, their rapid test with our reader, so that they lock the reader, they lock their chemistry, so that they lock their software, so that everything as a combo goes through the FDA. If you don't do that, then every time you change your reader, you have to go through the FDA again, even if your chemistry remains the same. And that's why you have to go to the developers and early on, help them go through the FDA process with your devices. That was essentially a very important part of the business, because otherwise the FDA created issues because, hey, you have 10 tests that went through the FDA, but hey, I cannot sell your tests with my reader because my reader is not FDA approved to read those. That's why early on, going through the FDA process would be very important. And that's exactly why we created that test developer software to help them get benefit from our reader to improve their processes and their repeatability, and at the same time generate data on how their systems work with patient specimen so that they can take that data and go through the FDA with it, right? That was the value proposition. In terms of the next steps, for paper-based sensors, I like multiplexing and I like more computational systems. So some of the work that we do in this space currently involves vertical flow assays. It's a sister of lateral flow tests. So let me show you a vertical flow assay somewhere here. For example, this one is a vertical flow test. It is the sister of lateral flow test. It's paper-based. It's 15 minutes of an assay time, but it's multiplex and the flow is vertical. It's still read with a mobile device, mobile phone or a Raspberry Pi or any kind of a mobile camera. But if you look inside, it has many immunoreactions put together. In this case, for example, you have 9 by 9. Every one of those colorful spots, that's one immunoreaction. That's one lateral flow test, quote unquote. So this one can do 81 of them in parallel. So what do I do with that? Well, I mean, it generates a ton of data for you. If you want to look at a panel of biomarkers for high-risk patients, this is great. If you want to diagnose a few conditions at the same time with some different biomarkers, different antigens on the membrane, this is great. We've been looking into cardiac biomarkers. We've been looking into COVID, immunity, IgG, IgM. We've been looking into Lyme disease diagnostics with this. Basically, bringing more sophisticated multiplex sensing capabilities to paper-based assays so that the readout gets more advanced, multiplex, look at a panel of diseases, biomarkers in the same time, 15 minutes, and with the same cost, dirt cheap, paper-based, a few tens of cents, and without changing the volume of the sample. 30 to 50 microliters of sample, sufficient. This is essentially what we've been working on. It's not commercialized yet, but this is something that I think would be very, very valuable for point-of-care diagnostics, home-based testing as well.